Hi guys, I hope you guys are enjoying your winter break. For my article, I wrote about the close-up views of tumors reveal new cancer biology. It is written by Philip Bale, posted on April 21st, 2020 of this year. And basically this article is about Philip Bale observing a couple of scientists. One main scientist is called Brad Bernstein, and Brad Bernstein, used a method called single-cell RNA sequencing. And what this sequencing did was observe each cell individually within a tumor to see which of the genes are active in that one tumor. And this method, helped him understand that cancer tumors, instead of only being one type of cancer cell, there are multiple cancer cells in that one tumor together that forms a heterogeneous mixture. And this mixture really set the tone of doctors creating medicine to help combat cancer cells. But as the article explained, these various types of cancer cells form in various ways, and they use various methods within our body to bypass our body defenses, especially the oncogene and the tumor suppressor gene, to survive and grow at a bad rate. And these cancer cells, one way they survive is by, even though they are non-malignant at the time, they are slowly growing, and they are using healthy cells to support their growth in a way. So these cells are not seen as bad, but they're helping these tumors grow, providing the proteins or whatever is needed to help these tumors grow. And another way these multiple cells are made is that for our stem cells, as we have talked about in class, stem cells are made to, once they are developed, that one cell is sent to a certain part of our body to function, like the muscle, or to our eyes, or to any type of part of our body to help function. It's committed to that one task. It does not alter from that one task or anything. But unlike that, tumors can differentiate from that stem cell, and this helps revert its initial outcome. The interchangeability of that cancer cell helps its survival rate. It makes it difficult for us to make the right medicine to attack it. And this way, these multiple ways that it can survive in our body, multiply in our body, is that even though the tumor suppressor gene tries to fix the DNA mistakes within our body and slow down the cell division, there are still certain cells that get past its defenses and mutates in other cells, which allows certain cells to grow out of control and lead to certain cancers. But for the oncogene, when there are too many cells present, as we have talked about in class, it would become a bad gene that would become active and lead to cancer. These certain ways, as we talked about, in class, these mutations in our body form in many different ways. And Brad Bernstein has really discussed about that and gave a more deeper meaning as to how cancer and tumor cells form.